Hello everyone, welcome to Chapter 2, Tissue Repair. In this chapter, I will introduce these contents: regeneration, repair by connective tissue, continuous wound healing, pathologic aspects of repair, and summary. Repair refers to the restoration of tissue architecture and function after an injury. Some tissues are able to replace the damaged components and eventually return to a normal state. We call it regeneration. For example, this is stomach wall. It has 4 layers: the mucosa, submucosa, muscularis, and serosa. This is a gastric ulcer with large area of tissue defects. The mucosa, submucosa, and smooth muscle are damaged and lost. After tissue repair, we notice that the epithelium shows complete regeneration, but the smooth muscle here is filled by fibrous tissue, which is incomplete regeneration. Cell regeneration can be physiologic and pathologic. The physiologic regeneration, which called cell renewing, such as gut epithelium, constantly partially lost and is replaced by cell division. The pathologic regeneration in cell injury, lost parenchymal cells are replaced by division of adjacent surviving parenchymal cells to restore injured tissues. In order to understand regeneration, we need to know cell cycle. Cell cycle includes interphase and mitotic phase M. The interphase consists of pre-synthetic phase G1, DNA synthesis phase S, the premitotic growth phase G2. Non-dividing cells are either in cell cycle arrest in G1 or they exit the cycle to enter a phase called G0. G0 is a phase that quiescent cells stay in a physiologic state. According to the cell's proliferatory potential, cell can be divided into 3 types: liable cells, stable cells, and permanent cells. The first type, liable cells, also called continuously dividing cells, it continues to proliferate to replace the destroyed cells and aged cells. Cell groups are stratified squamous cell, such as epithelium located in epidermis, oral cavity, vagina, and cervix. The lining mucosa of respiratory tract, gastrointestinal tract, the epithelium in the male and female reproductive tract, and so on. This type of cells retain continuous renewing ability because stem cells in the tissue have ability to differentiate into corresponding mature cell. The second type is stable or quiescent cells. In normal state, these cells don't divide or divide at a very low rate. In the situation of injury, quiescent cells will undergo rapid division. They are parenchymal cells in glandular organs such as hepatocyte, renal tubular epithelium, and mesenchymal cells such as fibroblasts. The third type, permanent or non-dividing cells. It refers to cells that has no capacity of regeneration. In human body, the nerve cells, skeletal muscle cells, and cardiac muscle cells are all permanent cells. Injury to brain or heart is irreversible and results in scar formation because neurons and cardiac myocytes have no capacity to divide. The damaged nerve cells are replaced by glial cells. The cardiac muscle is replaced by connective tissue. The following parts are self-study contents, including the nature and mechanisms of action of growth factors, the extracellular matrix and cell-to-matrix interactions, cell and tissue regeneration. Now, let's move to this part, repair by connective tissue. If parenchymal cells and stromal framework are all damaged, or if non-dividing cells are injured, repair cannot be accomplished by regeneration alone. Repair by connective tissue will play an important role. Repair begins within 24 hours after injury. Fibroblasts immigrate to the foci. Fibroblasts and endothelium of capillary proliferate. By 3 to 4 days, a specialized type of tissue we called granulation tissue is formed. In this picture, we notice there is a large area of tissue defect. It has a red, soft, granular appearance. That is how granulation tissue looks like. Under microscope, we can find capillaries. Like these circles, they are capillaries. A loose ECM contained lots of inflammatory cells, like this little blue spot. In this picture, we find many newly formed capillaries. They are here, here, and here. This cell is fibroblast. Here is another fibroblast. It's spindle with abundant cytoplasm and oval nucleus. In the background, there are numerous inflammatory cells like this one, the neutrophil, another neutrophil, also neutrophil. There are 4 steps of repair by connective tissue. Number 1, formation of new blood vessels. Number 2, migration and proliferation of fibroblasts. Number 3, deposition of ECM. The main component of ECM is collagen fiber. They are produced by fibroblasts in granulation. Number 4, maturation and reorganization of the fibrous tissue, or say, remodeling. Functions and development of granulation tissue. The granulation tissue can anti-infection and protect wound surface from further injury. It also fills in wound and the defect area. It can replace or encapsule the foci of necrosis, thrombus, and foreign bodies. The granulation will gradually develop to scar tissue in 1 month. Let's look at this diagram. As we know, the granulation tissue have 3 main components: the fibroblast, newly formed capillaries, and inflammatory cells. The fibroblasts synthesize lots of collagen fibers. Then the fibroblast will be surrounded by collagens and then gradually lost the capacity of synthesis and transform into inactive fibrocytes. Most of the newly born capillaries diminished. The rest of them are rebuilt into mature vessels. And those inflammatory cells finally disappeared. Thus, the granulation tissue evolved into scar tissue. Grossly, the scar is pale, stronger than granulation tissue, and has less elasticity. Under light microscope, it is composed of collagen parallel fasciculi with hyaline change sometimes, less fibrous cells, and less blood vessels. In this picture, we notice the large and irregular shaped scar on the leg. It is pale and protrude from the skin surface. The texture is stronger than neighboring skin. Under microscope, there are many wavy and parallel collagens and scattered fibrocytes, like this one. In this picture, the collagens undergo hyaline degeneration, shows a homogeneous pink appearance. This picture shows us overgrowing of scars. They protrude here from the skin surface and extend irregularly. We call them keloid. It is generally believed that this is related to the individual's constitution or the ischemia and hypoxia in scars, promote the mastocytes to secrete growth factors and cause the excessive growth of granulation tissue. Well, I will take the healing of skin wound as an example to show you what actually happened in wound healing. There are four main contents: inflammation, formation of granulation tissue and scar tissue, epithelial and other tissue regeneration, and the last one, wound contraction. The diagram shows us the wound healing phases. Inflammation begins the earliest, within several hours after injury. The time of granulation formation overlaps with that of inflammation. Generally, the granulation tissue fills in the defect area at the third day. From the fifth day, fibroblasts produce collagen fibers. After a month, the scar tissue is completely formed. The three pictures show us four steps of wound healing. Firstly, the tissue is destroyed, accompanied by bleeding and inflammation. Then, the neighboring fibroblasts migrate into wound zone. Here, in the second picture, the epithelial regeneration starts 24 hours after injury. The basal cells, here at the edge of the wound, begin to proliferate and migrate to the center of the wound, just underneath the scab, forming a single layer of epithelium covering the surface of granulation tissue. By 3 to 7 days, granulation tissue grow into and breach the defect. The epithelium proliferation continues, yielding a thick epidermal layer with mature architecture in the third picture. Collagens accumulate and remodel. Finally, collagen fibers are parallel to skin surface and the mature scar complete here. Based on the nature of wounds, the process can occur by first intention or by second intention. Healing by first intention, it occurs in clean, uninfected wound, such as surgical incision. Only focal epithelial basal membrane continuity is disrupted, and only a few epithelial and connective tissue cells undergo necrosis. Finally, the scar is small. A wound in the woman's neck here after thyroid operation. This is 2 weeks after operation. The wound is still red, and 2 years later, the scar completely matured. It became thin and pale. Healing by second intention. It is different from the first intention in 3 aspects. Firstly, the loss of cells or tissues is more extensive, or in the case of large wounds, ulceration, big abscess formation, the case after infarction in parenchymal organs. Secondly, the inflammatory reaction is more intense. Thirdly, there is abundant granulation tissue developed, and finally evolved to large scar. Let's look at these pictures. Healing by first intention. The wound is regular and small, with mild inflammation and smaller granulation tissue and smaller scar. Healing by second intention. A large clot and scab formed in the surface of the wound. Inflammation is more intense because of the tissue defect is large, containing great volume of necrotic debris, exudates, and fibrins. A large granulation tissue is formed, resulting in a greater mass of scar tissue. Within several weeks, original size of defect will reduce largely by contraction because of the presence of myofibroblast, which has contractile feature. Okay, now let's take a summary. We talked about cell proliferation, cell cycle, proliferative capacity of cells, regeneration and repair by connective tissue. We also talked about the pathological appearance of granulation tissue and scar. At last, we talked about healing by first and second intention. Here are some questions for you. Well, that's the end of this chapter. Goodbye.